Welcome to Dr. CM Edcast, your go-to resource for medical learning. Through discussions, case studies, tutorials, and debates, we help you learn, grow, serve.
Deep inside every one of our cells, there's a language of incredible elegance. It's the genetic code. You can think of it as a universal blueprint, the fundamental set of instructions that takes simple chemical information and builds the entire complex tapestry of life. So today, let's dive in and decode that very language. So here's the central puzzle, right? The language of our genes, our DNA, is written in an alphabet of just 4 chemical letters. How on earth can a script that simple build the staggering diversity of all life we see on this planet? Well, the answer lies in a truly remarkable process of translation. All right, so our journey starts in the cell's command center, the nucleus, which is home to our DNA. In there, a specific gene gets transcribed into a kind of mobile copy, a molecule we call messenger RNA, or mRNA for short. This mRNA then has to travel out of the nucleus, carrying those precious genetic instructions to the cell's protein-building factories. But, there's a catch. It's a language problem. The message on the mRNA is written in the language of nucleic acids, but proteins are built from something totally different, amino acids. The two languages are, well, they're just incompatible. So to bridge that gap, the cell needs a translator, a special kind of molecular go-between that we call an adapter molecule. And this essential adapter molecule, it's a type of RNA known as transfer RNA, or tRNA. Honestly, it's the unsung hero of making proteins. It's the molecule that's physically responsible for reading the genetic message and then delivering the exact right building block for that growing protein chain. So a tRNA molecule is a real specialist. Its one main job is to physically grab the correct amino acid and haul it over to the ribosome, which is the site where proteins actually get built. Its compact size, it's the smallest of the major RNA types, is absolutely key to its agility. This small, soluble little structure, usually just 73 to 94 nucleotides long, is just perfectly designed for its crucial delivery mission.
Okay, so the real genius of tRNA, it's all in its structure. Its unique shape is precisely what allows it to do 2 things at once: read the genetic message while carrying its corresponding amino acid. So let's take a closer look at its architecture.
Okay, so take a look at this. What we've got here are 2 ways of seeing the very same tRNA molecule. On the left, that's the flattened 2D cloverleaf model. It's great for helping us understand the basic parts. But in the real, 3-dimensional world of a cell, it actually folds up into that compact, L-shaped structure you see on the right. And that precise 3D shape isn't just for looks. It is absolutely critical for it to do its job correctly. At one end of this L shape, we've got what's called the acceptor arm. You can think of this as the cargo hold of the molecule. It always ends with the same nucleotide sequence, CCA, and it's right here that a specific amino acid gets attached, ready to be delivered. Now, on the complete opposite end of the molecule, we find the anticodon arm. This is the reading end. It's got this critical 3-letter sequence called the anticodon, and this sequence is designed to perfectly match or be complementary to a corresponding 3-letter word on that messenger RNA strand. So you see the brilliance here, right? One end holds the building block, the other reads the instruction. It's perfect. And all of this brings us to the code itself, the actual set of rules that runs the entire show. This genetic code is the dictionary, is the Rosetta Stone of life that allows the cell's machinery to understand exactly which amino acid is being called for by each 3-letter word in the mRNA. This table right here, this is the dictionary. Each 3-letter word, which we call a codon on the mRNA, corresponds to a specific amino acid. Let's try one out together. Say the codon is GGU. We find the first letter G on the left, the second G on the top, and the third U on the right. And boom, we land on glycine. Now, with 64 possible codons for only 20 amino acids, you'll probably notice right away that there's some built-in redundancy. You might be wondering why several different codons can all code for the same amino acid. Well, this is partly explained by something with a pretty cool name, the wobble phenomenon. See, the pairing for the first 2 letters of a codon has to be exact, but the third position has a little bit of flexibility, a wobble. This clever trick means a single tRNA can recognize more than one codon, which adds a layer of efficiency and even a protective buffer against certain mutations. Just like any language we speak, the genetic code isn't just a jumble of words. It's got syntax, it's got punctuation, really clear rules for starting and for stopping. And amazingly, it even has what you could call a few regional dialects.
The punctuation is super direct. No ambiguity here. The codon AUG acts as the universal start signal. It tells the ribosome, hey, start building the protein right here, and it also happens to code for the amino acid methionine. Then you have 3 stop codons, UAA, UAG, and UGA. These don't code for any amino acid at all. They are simply red lights that mean the job is done, translation complete.
Now, for a long time, scientists thought this code was absolutely 100% universal across all of life. And, well, it almost is. But we found these slight variations, these little dialects. And a really remarkable example is right inside our own bodies, in our mitochondria. For almost the entire biological world, the codon UGA means stop. But inside our mitochondria, the cell's powerhouses, that exact same codon is read as an instruction to add the amino acid tryptophan. It's a subtle change, but just a profound one.
So what we're left with is the system of just breathtaking precision. A molecular machine reads a chemical script and builds the very engines of life. And this process repeats, with near-perfect faithfulness trillions upon trillions of times a day, inside every single one of us. When you really stop and think about this incredible dance of molecules, it leaves you with a profound question. What does this elegance, this information processing at the most fundamental level, what does it really reveal about the nature of life itself?